So just to get then like that. Now we want to revise this paper, biology online. One, biology, kuna vitu kadhaa unafaa ujue. Ukirevise biology, itaki usome mara moja halafu umeacha kusoma. When I was in Form 1, nilipatanga exam ya kwanza kitu 55%. Exam ya ile kama kabisa. No, it was 70, then second time got 55. Nilikuwa nimejaribu kusomea. Then so nikarealize biology na several terms na inataka uandike kama ni binomial na nomenclature. So una mark on spelling. Any other subject ya science, mark spelling. But biology spelling. Like under new terms. Nikimaliza form 4 nilikuwa na hesabu terms zenye nimesoma za biology, just new terms. New terms. Those terms like mitochondria is a term that you have never heard in primary school, in high school. Biology literature, organelle, every glomerulus level complete. Including kind of performance. So to understand, unafaa unasoma, unarepeat kama maths. I hope maths kama unasoma mfanye tu Monday na Friday. Na ukisija yona mtu ameuliza swali ya maths yenye unashinda among. Hakuna ni ndani funga. Do revise it. Take your time. Revise maths like every day. Do just one math. Sawa. Mali tatu sana soma hii inaelewa. Soma, rudia. And that is also biology. So a cool house, the following affect energy requirements in the man, in your body size. Mtakona mwili kubwa na mwili ndogo. Who requires more energy? Mwili kubwa. Why? What about wakati kufanya mfano tunazungumzia about relative to your body like energy divided by your volume, like your size. So we talk about small animals losing more heat than nisema biology last week kwenye wafanya kujifanya mpakula kisema small animals. Let's say if the animal can equal volume 1, so 1.1 to 1 specifically. Bacteria ndio inatumika to lose heat from your body. Because your surface is important to the environment. Bacteria can, if it's more than 1 times 1 times 1, then times 6 equal to 6. So surface area is 6 centimeters square. So we have a surface area to volume ratio to 1 to 6. What about the double size? It's only 2. September. This one is 2. So volume is 2 times 2 times 2 equals 6 centimeters. 2 times 2 is 8. Surface area equals to 2 times 2, then times 6. 2 times 2 then you can have 6. volume ama surface area inakuja kwa inakuja 24. So volume surface area ukiweka surface area ya hii to volume ratio tunakuja 24:8, which is already 1. So surface area ya hii which means surface area na hapa ni volume. Which one has a larger surface area to volume ratio? Not the ratio, ni gani kwa surface area kubwa? So ni kama 1/6 na ni 1/1. So hii ni ndogo. Volume ndio tunadanganya. Volume ni kitu sorry. Hii ni andika 6 pabaya. Surface area ni 6. Hii ni 6. So we really got it for our one. So if you are compare and you can see the small animal. This small animal is particularly a bit more like. Therefore, to keep it warm, like tunasemanga joto la kwa tumbo.
Baila sai.
Baya.
Sasa sote nikisha kutoka kwa shule yao.
Kuna ya saa 6 hawa ni form 1.
Sasa nikisha hivyo na mimi brother kadogo hapo.
Wewe unafanya business?
Mimi sasa bila kompyuta.
Unataka kuja au kompyuta?
Sasa unakuja Canada hizi ndio mimi kompyuta.
Alafu tutakuja saa 6.
Vijali lala.
Sawa.
Kama nikutumia pesa?
Si kuona lakini nitaangalia. Si kuona.
Tunamuzika.
Sawa. So we are revising from one biology, the class that you did. So you can check the class that you did. The class we did on Saturday or that I sent online for Form 1. So we are discussing the first question as explain how the following affects energy requirements in mammals. So one of the factors that affect energy requirements, apart from now taking your in terms of volume energy, yes, a large animal will take more energy. Lakini tunasema energy versus volume. So ni kama iseme rats. Uchukue rats kilo kama elephant moja tuseme iko na 1000 kg. Alafu uchukue rats nyingi upime zikiwa 1000. Alafu sasa ujiambie uangalie ni nini nakula chakula nyingi. Hizo rats 1000 zitakula chakula moja. That is what we do. So unaweza andika you have red pen or black. You can hapo juu. Unaweza andika smaller animals. So question 1A, smaller animals have large surface area to volume ratio. Smaller animals have large surface area to volume ratio. Therefore, they tend small animals tend to have a large surface area to volume ratio. Therefore, they tend to lose more heat relative to large animals. Relative Parentheses to large animals. To large animals. Therefore, they will need more energy to maintain body. They will need more energy to maintain body temperature. The second question asks about being male. So, males and females do require more energy. Why? Na hata wasipofanya kazi, tuseme tumekaa tu like sisi tunasoma, more masculine, more muscles means they need more energy. Muscles itself inatumia energy to respire. Ama when watu kama binadamu unapata muscles zake zinahitaji more energy. Now, tunaandika males require more energy than females. Males require more energy because males require more energy because they have more muscles. They have more muscles. They have more muscles, which require energy in respiration. So require more respiratory energy, require more energy in respiration. Hi. Name the branch of biology that deals with the study of the following cells. Psychology. Development of techniques for application of biological processes. Biotechnology. Structure of tissues. Histology. Arrange the following in the ascending order. So what are we arranging? Tuna kingdom, so so in the ascending is mentioned again a small, large. Species, then class, then unajua stuff ya physics. You're supposed to say, andika somewhere, sijui even in your book, ama wherever tunapata andika physics. Philip. Ziko zimeteremka. King Philip can only find green socks. Ndio useme king ni kingdom. King Philip can only find green socks. Kama sisi tu. King Philip can only find green socks. So you put kingdom. Film. Order. Family. So tunaanza hivyo hivyo, ni fawaja peana zote, unakuja tu unajua kuanzia chini usikishi. Then order. Then hiyo film from division. Film ya animals, division ya plants. Kila form green tunasoma classification topic 1. Kila short data. Hiyo cell, organ, organism, organelle, tissue. Ile kidogo kabisa yenye unafikiria, ile kidogo kabisa unadhani ni nini? Kwa hivyo ukachakata kidogo ili tutafuta kitu ile kidogo kabisa utapata nini? So we are in question 3. Question 3B. So we are in question 3B. Arrange the following in ascending order. Division hiyo tumesema unatumia King Philip can only find green socks. So king is for kingdom, then phylum or division. C is for class, then there is order, family, genus, then species. Now we are in 3B. Now, this is the Form 1 cut that we did last Saturday, last weekend. So the smallest in cell, organ, organism, organelle, and tissue. Unadhani ni? Organelle. Haya, organelle, example ya organelle. Tunasema humans ama animals or even plants are made of cells. cells are the smallest functional unit in a living organism. Now that smallest functional unit kama human cell, ndani yake kuna organelles, kuna mitochondria, kuna plasma membrane, kuna hizo stuff zote. So the organelle is the smallest. Then sasa tunaenda kwa cell. From then the cell tunakuja kwa tissue. Muscles are tissues. So you have a hand, but the hand has muscle tissues. Then from that tissue you have the organ. Your mkono ni organ. Ama your heart is an organ. Your eyes are an organ. Then from an organ we have the whole you, which is an organism. Okay? So the smallest in that order, the smallest is organelle, cell. From the cell we go to tissue. From tissue we have the organ. Then from the organ we have the organism. So we are revising biology from 1. Question 4 reads, a normal plant cell was placed in solution X and went through stages P, Q, R, then S. So this is a normal plant cell. Remember these stages. So study it well and see what has happened. So what do you think has happened? Kuna change imefanyika mahali? Change gani hiyo? Cement water. Kwa cell sap. Water in the cell sap. So cell sap of the cell ndio hii hapa. Imeenda ikikuwa ndogo kutoka ikubwa. Then you can also see another change that has happened is that the cell membrane. Plant cell iko na cell membrane na bado iko na cell wall. So the cell wall has remained intact, but the cell membrane has retracted from the cell wall. Kunaanza kuwa na space, ndio hii inabaki space na ndio hii yote. So in that case, state the identity of solution X. So where that plant was placed in that solution, ndio ikalose maji. That solution was hypertonic or hypotonic. Mumesoma hizo? Ndio. Hapo kwa highly concentrated. What are you written? So you are supposed to say something like that. So it was, the solution was, sorry. The solution was? Hyper. So the solution was hypertonic. Ndio maji ikakuwa lost from the plant cell. So where this cell was placed in the solution that this cell was placed into was hypertonic, so that it lost water. So hypertonic. The other question is, account for the appearance of the cell at stage S. So account, you explain kwa nini inakaa hivyo. So you explain kwa nini the membrane ime-retract na kwa nini cell wall ime-remain. What are you supposed to say? So you're supposed to say, see, tumesema maji imelose your maji. So you're just supposed to say that in English. So this is what you say. So when the cell was placed in a hypertonic solution, when the plant cell, another cell, because we are talking about plant cell, was placed in a hypertonic solution, it lost water through osmosis. The plant cell lost water from its cell sap. So it lost water from its cell sap. It lost water from its cell sap through osmosis. Cell sap to the solution, through osmosis. So the plant cell lost water through osmosis. Plant cell lost water from its cell sap to the solution through osmosis. As a result of shrinking of the cell sap, shrinking SHRINKING. As a result of shrinking of the cell sap, the cell membrane retracted from the rigid cell wall. So retracted from the rigid cell wall, which cannot shrink, which resists shrinking. So ndio maana the shape and the size of the plant cell haipungui vile, but the plasma membrane ina retract. Sawa? So it's good you understand, unaelewa nini. Haya, so question 4, part C. At stage S, the space between the cell wall and the plasma membrane of the cell was found filled with solution X. At stage S, the space between the cell wall and the plasma membrane of the cell was found filled with solution X. Name the physiological process that enabled this. What do you think? What happens so that your solution will come up? You may get through what process? Crenation. I don't think so. Mostly your movement ya hizo waters and stuff inafaa kwa either osmosis, diffusion or transport. So maji ilitoka na osmosis kwenda hivi, but hii plus 100 tunasema it is selectively permeable. What about here? Because maji inaonekana inaingia tu virahisi. Turgid pressure inakuwa nini? Turgidity. I think maji can move up through diffusion. It will just diffuse from there. Which let me eka hiyo scan tutakonfirmia what will happen. But unaona kuna kitu tumesema inaitwa crenation. That is I'll just confirm. Inapita through physiological process must be diffusion, osmosis or active transport, which I think is diffusion. Your magic will get. So because I'll just confirm. So we put a star on 5C to confirm whether it's diffusion or osmosis. Sorry? 4C. Oh, 4C. Inasema 5. Question 5. Inasema Samson wrote the scientific name of lion as Panthera leo. Give the name of this system of naming organism. Scientific naming ni binomial nomenclature. Sawa, binomial nomenclature hiyo ndio system ya kupea animals or plants two names. Okay? So Panthera leo, question 5A. It is binomial nomenclature. 5B. Write the scientific name of lion in the correct way. Andika Panthera leo. So make sure L is small na P ni capital. So the correct naming unafaa Panthera, unaandika P capital and you underline separately kwa pamoja. There. So you're supposed to write this way. Capital P, small l. I don't know whether they are inverted on your side, but you are able to see whether P is capital and how they are underlined. And I can show you this. Panthera leo, like that. So first it was wrong. Why was this naming wrong? Because alikuwa ameandika na capital letter, sio Panthera leo hapa. Alikuwa ameandika. This one was written in capital letters. So you're supposed to write in small letters. Only P is capital and L is small, then you underline separately. But when you're writing in computer, inakuwa italics, sio? You just write in italics. But P again is capital, L is small. So, question 6. Penina counted 10 onion epidermal cells along the diameter of field of view of a light I've forgotten to bring the camera back. Penina counted 10 onion epidermal cells along the diameter of field of view of a light microscope. The field of view had a radius of 2.5 millimeter. Determined the diameter of one cell in micrometers. So what are you supposed to do? What did you do? You left it blank. What did you do? You're taking the notes. Can you... So, have you seen confirm from your notes to how many micrometers are there in 1 millimeter? 1,000. So what you're supposed to do here, so field of view inakuanga circular. Sindio? Alafu yeye ameona ziko 10. Amehesabu 10 cells. Radius hapa, radius ni mpaka katikati is 2.5 millimeters. So what's the diameter? Diameter should be 2r. Radius ni hapa katikati. So you're supposed to multiply by 2 to get the diameter. Diameter kwa maths. What have you done here? In your notes. During a practical, the line of one cell that the field of view was. Hii ni tofauti. This is 8 millimeters wide. Sawa. So unazihesabu, unachukua if this is kama hii yote hivi length yake ni 8 millimeters and you see wind diameter, width of one cell is diameter of a number of cells. Na sio kitu unafaa kukram, like when you know this distance and you know how many cells are here and assuming they are equal, you are supposed to divide. Okay? So we are talking about the field of view which is circular. You are supposed to field of view. The radius we are given is 2.5. Is it 2.5? 2.5 millimeters. So you are supposed to find the diameter, which is 5 millimeters. Then how many cells were there? 10. So I want you, with that information in here, the width of one cell. Determine the diameter of one cell in micrometers. Just first, kwanza in millimeters, then we will convert. Nimewapea hiyo, nimewaelezea hii yote ni hapa nusu ndio 2.5 radius. So the diameter the width, how wide that field of view iko ngapi? Unafaa by 2. Ukishapata hiyo na hapa uko na 10 cells. So swali yangu unaulizwa ni how wide is one cell? How wide is one cell? How wide is one cell? Let me also go here. So you have your field of view. Field of view. this is your field, and you have counted 10 cells like this. So, don't mind when you're toshani, assume zinatoshana. And you have been told the diameter kutoka hapa up to here. This one is kutoka hapa mpaka hapa ni 2.5 millimeters. 2.5 millimeters. $2.5 millimeters. So this is 2.5 millimeters. So what about the length of this wing? If this is 2.5, what about the diameter? So the diameter is 5. So the diameter here is 5 millimeters. And here you have counted 10 cells. You have counted 10 cells. So meaning up to this point you have counted 5. 1, 2, 3, 4, and 5. Pande ingine kuna 1, 2, 3, 4, 5. We are being asked about the diameter of one cell. So what are you? You say radius is 5, then? Oh yeah, so you can convert that 5 millimeters into micrometers. Multiply by 1000, they'll give you 5000 micrometers. Then you divide by 10. So this wind, you can 10 cells inside them. So you're not supposed to multiply, you're supposed to divide. Kama hapa ni 5000, then hapa pekee inapakwa kidogo. It is 5 divided by 9. So after you have understood, now you can write your formula. Wind of cell is diameter of, no, see your diameter of cell, diameter of field of view. Field of view. Hapo ndio kuchanganya. Unatafuta diameter of field of view. So you can write that formula. I think you have understood. Wind of cell stroke diameter of cell is given by diameter or wind of field of view, field of view divided by number of cells counted. Kwa maana ni hesabu kidogo sana. Sasa ikuanguka hiyo. Okay? Yes, you should not fail. Wind of cell. Tafuta hizi zingine zenyewe. So that's how we have it done. And now we continue with the questions we have been doing. So question if you do that, you see diameter is 12, diameter is 5 mm times 1000 will be 5000 micrometers. Diameter of cell equals to 5000. Tumesema diameter of field of view over number of cells counted. So is equals to 5000 micrometers over 10. Should be 500 micrometers. So if she used eyepiece lens magnification of times 15 and objective lens magnification of times 40, determine the following total magnification used. So if she used eyepiece lens magnification times 15 and objective lens magnification times 40, determine the following total magnification. Total magnification means yes, on account of 10. I can see your millimeters. For units, you just cancel the units. And you give yourself so total magnification is equals to times 15. Total magnification is the multiple, a product of magnification of eyepiece lens and magnification of objective lens. So times 15, then multiplied by times 40. You get times 600. No, times. Yes, times 600. Actual diameter of one cell. Actual diameter of one cell. So when the cell was magnified, it was seen to have the diameter of 500 micrometers. So what is the actual diameter of one cell? It's already magnified. When it is magnified, it is 500 micrometers. 500 divided by 1000 is a half millimeter. You know millimeter from your ruler. Hiyo side ya inches unakuwa na millimeter. Hapana, that side ya hiyo side ya centimeters. Chini ya centimeters unakuwa na millimeters. Sawa. So with our calculations inatuonyesha nusu ya hiyo millimeter hapo ni cell. You have a ruler. This side ya centimeters ni kwani ni zote? Hapo ni centimeter tupo. So uto twenye tunaonekana rula inakuja class ya biology. Mko sawa? So wanasema hiyo cell ni nusu ya aka moja. Na inafaa kuwa microscopic. Sasa ikiwa magnified ndio inaonekana ikiwa hivyo. Yes. From centimeter side. So what you're supposed to do already kwa magnified, tunafaa tufanye to multiply ama to divide. So actual diameter is given by length ama diameter of magnified cell. Magnified cell divided by total magnification. magnification. So inafaa ikiwa 500 micrometers over 600 ndio ikupe 5/6 micrometers. Kuna mtu ako na calculator, so unaweza andika hiyo 5/6 au divide 50 then 6 0 like a 0 here. So 6 goes in 50 how many times? 8 because of 48. No. Yes. 48. 2. Then it goes 2 times because of 16. 34. Then like a 0. Inakuja 6 times. So it is 5/6 micrometers, or but then there's another 5/6 micrometers, or if you divide it and prepare 0.82, acha tuone kama kuna 36. So from there kind of ikuanga 6 nyingi hiyo. So itakuwa baada ya 26 unaandika 27 micrometers, 0.8. So the answer for, I hope you have understood question 6. So the first question, part A, ilikuwa nauliza the diameter of one cell in micrometers. So you find the diameter of the field of view, you divide by the number of cells you find there, or you see. Very easy. Then total magnification, you multiply magnification of the eyepiece lens and objective lens, because hii ina multiply by 15 alafu ina multiply by 640 inaendelea kuwa magnified. Okay, then actual diameter of one cell, unachukua ile total magnification na ile the diameter of one cell. Unachukua the diameter of one cell, una divide by total magnification. Because it has already been magnified, you want to see the actual. So you go back. Okay? And I'll tell you something. Give 2 reasons why this method of cell size estimation is not accurate. So give two reasons. Give 2 reasons why this method of cell estimation is not accurate. Because
they use the
light microscope, which so they use light microscope rather than an electron microscope. A light microscope has low magnification power compared to an electron microscope. So hiyo ni answer moja. So one possible source of error is that light microscope has lower magnification power than electron microscope. Therefore, kuna some cells zenye zinaweza kuwa hazionekani. Sawa? So, light microscope has low magnification power. Therefore, some cells could have not been counted ama seen. Kuna hiyo risk eh? Haya, the other one ama another, unaona cells mbili ni kama moja, unaona? Another one is that light microscope introduces human error. Introduce human error because wewe unaangalia na jicho lako na unaangalia. Electron microscope inakuletea kwa screen, inaangalia then inaleta kwa screen, so which is easier. Hata inaweza hesabu ikwambia how many cells are there. Sawa. Most of modern electron microscope are automated. So what you say is that light microscope introduces human error. So that's the second answer. The first answer, of course, was light microscope has low magnification power. Kuna swali kwa hiyo. So this question yenye nimewaletea hiyo kwa zinakuja kwa paper 2. Unaona ni 8 marks. Nimetoa mahali ya 8 marks. Inaweza kuja KE ikiwa hivyo. Inakujanga structured question on one topic, wanauza like small issue with the internet. So name 2 we are asking. So you are saying that Q expand. So name 2 materials in the school laboratory that can be used as R. One is visking tubing. Another one is alternative to visking tubing. Serpent skin. Serpent kama ile ya nyoka. Kuna kitu inaitwa serpent skin na behave kama cell wall, so it is selectively permeable. Sawa. Serpent skin tubing. So for question 7A, we are saying the alternative to visking tubing is serpent skin tubing. So 2 materials in the school laboratory that can be used as R is visking tubing. R you can see either visking tubing or serpent skin. You can see better here. Visking tubing or serpent skin tubing. Then, which term is used to describe solution P in relation to solution Q? Now that you may expand, see your solution P ilikuwa use the term in similar terms like what I had given you. The term used to describe solution. So not wakati unaulizwa describe solution, unaona mmeandika the same thing lakini ni wrong. Nilikuwa nasema watu na formation. You cannot write the wrong thing na hakuna formation nyingi wote. So when you osmosis ni physiological process. Osmosis, active transport and nini? Osmosis. Active transport. Hiyo zote ni physiological process. But when we are talking about term used to describe a solution, you are describing that solution in relation to the cell surface of the solution inside the visking tubing. So ndio maji ingie kwa that visking tubing, inamaanisha solution ilikuwa na high concentration ama low? Solution outside. Solution ilikuwa high concentrated. Nje. Tunasema maji, chumvi, tuseme chumvi ndio imekuwa hapa ndani. So na pia kuna chumvi, lakini moja ni kuna chumvi nyingi. Ni gani kuna chumvi nyingi, hii ama hii? Ukiangalia hii ndio maji iweze kuingia ndani. Si maji na mtu ala chumvi kwenye kuna chumvi nyingi ndio itaenda. So inamaanisha hapa ama hapa ndio kuna chumvi nyingi hapa ndani. So inside the visking tubing there was high concentration of solids. Iko na chumvi nyingi lakini, so ukiweka maji chumvi nyingi inamaanisha maji inapungua. Sawa? Unaelewa? So there is high concentration of solids. So water moves inside the solution with high concentration of solids. Sawa? Ama you can say maji ni nyingi hapa, so inaingia huko ndio iende neutralize. So water is highly concentrated here, but solid, your salt, iko highly concentrated here. So water moves from where water is highly concentrated to where kwa solids ziko highly concentrated, but water less concentrated. I hope that one does not confuse. So inaingia ndio ina expand. So now that this one, in terms of your solid concentration, we can sema hypertonic, hypotonic. In terms of hypertonic and hypotonic, it means which solution, the solution, so that water moved inside the viscous tubing, the water is highly concentrated. The water has high concentration of solids or less. Ni gani kwa more concentrated na solids ndani? So now we are talking about water. So water hapo nje ni low concentration. So low concentration inamaanisha hypertonic kama hypotonic. Hypertonic or hypotonic? Hyper means high concentration. Hyper. Usengi mtu akiambiwa wewe uko hyper, uko na moods, unaonekana uko hype sana. Hyper. Hypo ndio low concentration sana. So ni hyper ama ni hypo? Hebu angalia. So we say hii iko na less, iko na chumvi kidogo, hii ndio iko na chumvi nyingi, hii ndio hyper. Kwa hiyo hii ni hypo. Sasa swali liko nauliza ipi? Nimesema kusema hyper. So you are supposed to say hypotonic. So the solution, which term is used to describe the solution? P in relation to solution Q. Unaelewa? In relation to solution Q. So this one in relation to Q itakuwa hypotonic. So kalamu yako nini inafanya? It is hypotonic. Na ikiwa zinatoshana inaitwa hamjasoma? Kuna kitu kama mesotonic. Kuna kitu kama mesotonic. Let me just confirm for you whether it is hypertonic. Just a moment. We check whether is it mesotonic. A solution is not. If a solution is not, you can also write somewhere hypertonic, higher concentration than higher concentration. You compare with another higher concentration. Hypotonic, low concentration. Isotonic solution. An isotonic solution is one that has equal concentration with the other compared solution. If you're talking about so next time we will listen how to describe solution Q in relation to P. Utasema, is it hyper, iso, or hypo? Now we are talking about Q. This question was talking about P. P in relation to P. Now P ni hypotonic. What about Q? Q ni hypertonic. Ukipata isotonic, these will not change. Sawa? Next time utaletewa na mwonyesho imerudi mpaka hapo. Same. Utasema that solution was isotonic. Okay? Account for the observation made in Q after 1 hour account. Now, hapa tumechorewa after 1 hour vile inakaa. Na ni 3 marks. So hapo uko na time ya kusema what happened when you explain why. So what happened in solution E after 1 hour? After 1 hour, what happened and why? I'm giving you time first to process that. No control C, high concentrated. No. Halafu in brackets hypotonic. So tell me what happened in your own words. Solution P, which had a high concentration, moved into the solution. Remember what moves, It's not that solution, it is the water. It is not the whole solution. Water only. So talk about water, don't talk about solution. Solution P, which had a high concentration, now you are wrong because it's not high concentration, haitaenda. Mnashanganga kwa nini tunasema ati osmosis movement of something from high concentration. Haya. We are talking tunasema hivi. So if you have let me demonstrate here. So you have this. So this one, tuseme hii maji inatoshana. Maji inatoshana. Then I don't know why. So this is water. Ukiweka hapa one spoon of salt. This one. In terms now salt, tuseme ndio solute. Solute ni salt, maji ni solvent. Sawa. Okay? So, which one is more concentrated if you are describing the two? What is concentrated? Solute concentration is high. Sawa. When you're talking about hypertonic and hypotonic, we talk about solute concentration. So this one is hypertonic compared to this. But when we are talking about water concentration, which of these two has high water concentration? This one. So, but when you're talking about water concentration, hakuna hypertonic kama isotonic. Hypertonic haita change anything. Hypertonic we talk about solute. So water moves from region of high concentration hapa kwenye hakuna chumvi inaenda huko. Sawa? But when you are talking about kama ni chumvi ilikuwa inaenda Unajua hapa hii ni semi-permeable. If ni chumvi ilikuwa ina move, chumvi would move from high concentration to low concentration through diffusion. Unaelewa kama? Unaelewa? Because you are laughing. We are saying, this is water, this is water, hii ni chumvi. Water imewekwa chumvi. So when we are talking about concentration, we are talking about both water and other particles. When you're talking about diffusion, diffusion is the movement of particles. Can be molecules, can be anything, but not water. Moving from a region of high concentration to a region of low concentration. So, you know, diffusion na osmosis zote zina move from high to low. Sawa? From high to low. Lakini active transport moves from low to high. Low to high. But osmosis ni water. Hizi mbili ni can be particles, ions, molecules, vitu nyingi. Sawa?
So ni maji kingia ndani osmosis.
When water inaingia kwa ingine ni osmosis.
Hii kutoka ni diffusion.
Ni chumvi. If ni chumvi inatoka ni diffusion. But remember, hiyo distributing ni selectively permeable. Ni kama cell membrane. Chumvi ingia. So chumvi haitaingia. Inavuta maji kwenye chumvi iko. Chumvi kwenye iko inavuta maji. Sawa. Haya, lakini in other cases uambiwe uchorewe kama hivi, alafu wakaweka membrane kwa hivi. Sasa ni solid pekee ina move. Or tunaambiwa hivi na harufu ina move through diffusion from something that is dead to here. Ina diffuse kutoka kwenye iko highly concentrated to here to low concentration. Ina move. So hiyo inakuwa diffusion. Haya, when mmea ina absorb mineral salts inatumia diffusion, because mineral salts they are like input solids kama fertilizer kwa mineral salts. Sawa, ina diffuse, then ikikuwa kidogo, when there is low concentration of those mineral salts in the soil, now they start moving through active transport. Ziko kwa hii mimea. So this is soil. Now, ishavuta hapa tumebaki low minerals. Sasa kwa plant cells ndio kuna low minerals. So lazima plant itavuta hizo mineral salts through active transport. There is low concentration to high concentration of mineral salts. Lakini actual cell in most cases hapa ndio kuna high concentration ya hiyo mineral salts inapovuta. Sawa. So mineral salts are absorbed through diffusion and osmosis. I think iko kwa hiyo kata last time. Nimekumbuka. Wakati ulifanya kisha. Haya. So umeelewa. So in this case we are talking in terms of hypertonic and hypotonic. Water will move from hypotonic to hypertonic. Water will move from hypotonic to hypertonic where there is high solid concentration. Water will move from here to here. So, lakini inamaanisha tumetoka from a region of low concentration hapana. Ni where water is highly concentrated to where water is lowly concentrated. I think we have understood that concept. Now, account for the observation made in Q after 1 hour. So you write water moves from hypotonic solution P. Water moves from hypotonic solution P. Hypotonic solution P, where water molecules are highly concentrated. Water is from hypotonic solution P, where water molecules are highly concentrated. Water molecules are highly concentrated. Two, highly concentrated through as an impermeable membrane. To solution Q. Impermeable membrane to the hypertonic permeable membrane to the hypertonic solution Q. Hypertonic solution Q, where water molecules are in low concentration. Water molecules are in low concentration. Water molecules are in low concentration. Causing volume of solution P to increase. Volume of solution Q. Volume of solution Q to increase. Haya, so hiyo umejibu. Kuna mtu alikuwa ameandika I think kwa pepa yao wengine that solution P will decrease. Utaiona kweli? If you don't remove this, will you see that it will decrease? Hiyo volume, even if inaingia, hii volume will remain the same. Hiyo level. Because ina increase, ni kama ukirusha mawe ndani ya maji. Ukiendelea kurusha mawe, hiyo level inaendelea hivyo. But now in this case, hii maji inaingia, kwa hii level yes increase. Hata hiyo ongezeke aje. Level hapa iwezi increase. Maji imetoka hapo, imechange tu mahali ilikuwa. But if you remove and measure, you can only increase. But from what we see, solution P itapungua yes, lakini that level hautaona. You will talk about what we observed. Seven D. How is the process investigated in the experiment shown above important to the function of chloroplast? Nimesoma about chloroplast. So hiyo process inasaidia chloroplast trap light energy for photolysis. So how is that process important? Chloroplast How is that process important for chloroplasts? Chloroplast kazi yake ni photosynthesis, right? So it requires what about stomata? Cell yenye inafungua stomata inaitwa stoma. So how is osmosis important to chloroplast? How is osmosis just So, mumesoma about photosynthesis. Nutrition in plants. Photosynthesis. Kuna light stage na dark stage. Light stage involves the splitting of water molecule. Then dark stage combining that splits water molecule with CO2 to form glucose and CO2 and oxygen. CO2 in a combined plants will absorb water. Chloroplasts absorb water from adjacent cells. Chloroplasts absorb water from adjacent plant cells. Adjacent kumaanisha zilizo karibu nayo. SC, adjacent SCE, adjacent plant cells, plant cells, through osmosis. Water is required in photosynthesis. Water is required in photosynthesis. And it is combined with the CO2. It is combined with the CO2 to produce glucose and oxygen. Oxygen. See, plants are in emit oxygen. They take in CO2, they produce oxygen. So, mkienda kwa ng'ombe, unaweza andika glucose na kamba. H2O plus CO2 in a pair, C6H12O6 plus O. So, turudi unaandika kama hii ni water plus CO2. Sasa tunaenda kwa mtu. Tunaanza kusoma hivi. Glucose ni C6H12O6. Equal to CH, H2O, H na 2, H na oxygen, hydrogen and oxygen zinatoka hapa. Na pia hapa kuna oxygen. Carbon ndio hii. Then na NH2. Now, if you want to balance, hapa hizi ni sita, unaweka hapa sita. Alafu hii ni 12, unaweka sita, ndio tena hapo iko 12. Hiyo iko sawa. Then oxygen hapa ni 12, because ya hii 12, na hapa ni 6, na hapa ni unaweka hii. Unaweza kama hiyo. Katika this people. Ini namanya kalau kita takut, kalau kita semua kali kesal. Now, management now let me take the question. Hawa wengine nione ama kuna kitu relevant kwenu. We have finished revising the exam unit. And I've seen you have an issue with the osmosis and diffusion. How do I find again? Technical exercise, the excretion and secretion. Oh, really sorry, the kid of a long of times. Right? Really? Does it for some? I was wondering. Haya, andika hii swali. Do you need a break? Tumesoma like 1 hour 30 minutes. Do you need a break? Tunafaa tumalize 11:30 na sasa 11. Tunafaa break ya 5 minutes alafu tusome 25 minutes. Sindio? Dakika 5 ni za kutoka hivi kuruka kwenda washroom na kurudi. So we take a break and we will be back the very right now. Tusha take a break. Na lazima mtoke, tokeni nje. You relax. So we are on a break for 5 minutes. We are going to be back. I'll not put off the camera, so itaendelea tu kuwa hapo while we are on break. Just 5 minutes.
Okay. See that same as
We
this
are
one.
on break.
We are on.
So yeah, come back. So we are assuming our class, biology class. So unajua mmekaa mpaka 11:20 kama tungeenda hiyo break, but it's okay. So we are going to do something in the next 15 minutes.
Desiree.
Sorry?
Desiree. Sorry for that. So she was nana kirabu ayali na kuna vile na feel ya. So nilikuwa na reaction yangu akaanza kucheka. Sasa mimi nikasema kicheka sio tu
Oh. Oh, ni sana. Thank you for clarifying. I was wondering kwani kuna shida na mavazi yangu. Oh, so you don't like feeling a sauti? Comment or just? Oh, sad. Now write this question. Aka can move. Aka can move. Can move. Can move, uses oxygen to produce energy and CO2, carbon dioxide. What else can it do, like living things? So, what else? Kama hiyo ya energy na oxygen. Akija hapo useme, why is it not considered a living thing? Car. So why is a car not considered a living thing, not a human being, a living thing? So why is a car not considered a living thing? It is taking in petrol. Yeah. So if it is nutrition, it is taking in petrol. So car that does not have growth, it cannot grow.
So as a teacher today, the simple question: how much is it called move using energy to produce carbon dioxide and water? Similar characteristics occur in living beings, and yet the vehicles are not classified as living organisms. They explain because they do not reproduce. Nor does it experience gaseous exchange or its ability to produce organisms have this. You are the boss.
So, hiyo swali, umeona hiyo swali venye naulizwa, that's the question I wanted to phrase. So what you say, hiyo ya gaseous exchange hapana. Mliambiwa gaseous exchange? You know, si kwa swali imesema it produces CO2. Kama inachukua oxygen, hiyo respiration huwa inatumia oxygen. So hiyo ya respiration pekee ndio sio answer, but it cannot reproduce, it cannot grow. Unasema si kuna kama na 8 characteristics of living things. So you just list the other characteristics that are not seen in a car. Ukitaka kujua car huwa ina respire, inatumia oxygen, you go in a closed room like this and you leave a car engine running, alafu funge mlango and everywhere, it will consume all the oxygen and release CO, carbon monoxide, because kuna less oxygen supplied. Sasa hiyo, it is poisonous, can kill someone. So there are some people who have died in a poorly ventilated garage. So ikona respiration. And if respiration inamaanisha hiyo consumption of oxygen to produce energy and CO2 iko pia. List one roll of Golgi bodies. One roll of Golgi bodies. One roll of Golgi bodies. So we have answered why a car is not a living thing. You list other characteristics. We have 8 characteristics of living things, and we can just say them. Nutrition, respiration, irritability,
Growth and development,
Yeah. So what's the difference between gaseous exchange and respiration? What's the difference in your own words? What do you think? Where does gaseous exchange occur? In the? Wapi sasa? Which organs? Kwa lungs, gaseous exchange. In a plant, kwa leaves, sindio? So where does respiration occur in a human being? You can't remember? So respiration is breaking down of foods to produce energy. So inafanyika kwa cells, kwa muscles. Sawa? So that is now respiration. Kaa ina produce energy na pia inafanya gaseous exchange. So if it is nutrition, it is taking in petrol and it is also moving. So hizo ni characteristics kaa nne. Gaseous exchange ni moja, respiration ni nyingine. Hiyo producing energy ndio kaa move. Hiyo ni energy. Movement ni nyingine. Na if it is nutrition, ina take in petrol. Hiyo ndio inakula. It is consuming na inaisha. So ile yenye haiko ni reproduction, growth, irritability. Gari ukiguza ziko na alarms siku hizi.
But
si zote. Irritability ni sensitivity. Gani nyingine moja tumeacha? Reproduction, sensitivity ama irritability. Growth, like it. So kuna reproduction, growth, irritability na gani? Jaandika mahali. Na? You have a question? Tunamaliza in the next 5 minutes. Ndio class inaanza saa 6. Unaenda saa ngapi? Na saa hizi ni 11:30. Unataka kwenda home?
Hadi niweze kufungua.
Unataka kwenda wapi?
Niko tu kwa around. Sasa ya kula watu.
Just around wameenda lunch. Okay. So I want that other characteristic. Oh yeah. Ina remove waste. Hadi gari ina remove waste. Hiyo moshi ndio gaseous exchange. Ni part of excretion. But kwa gaseous exchange kuna hiyo moshi kwa exhaust. Ukisoma topic ya excretion ya form 2, kuna hiyo part yenye huwa tunasoma about lungs na wake yake na excretion. So not gari zile characteristics aina ni growth, reproduction na irritability somehow ukikosa tu like tu, but growth and reproduction sawa. Haya, so role ya Golgi bodies, you have your notes za form 1 confirm. Every exam lazima ikuwe na moja moja. Roles of Golgi bodies or Golgi apparatus. Their shape ni gani? Halafu andike role ya mitochondria. So which organelle is not present in an animal, a plant cell, but absent in an animal cell? Which organelle of a plant cell or animal cell? I think I should give you that as an assignment. But sorry? Subvacuum. Haiko kwa animal cell. Ya pili, cell wall. Plant cells do not have a cell wall. Umeandika hiyo wama ito. Umeandika hiyo that cell wall and subvacuum are only available in plant cells and not animal cells. Umepata Golgi bodies?
They acknowledge and transport the proteins and secretions.
So, transportation and packaging of glycoproteins and secretions. So, in function of Golgi bodies, packaging and transportation of glycoproteins and secretions. Packaging and transportation of glycoproteins. Glyco, you know the spelling. Glyco. Hata kuandika utake ama utake sijui ama blackboard zinaandikanga hivi juu hii inanishi. It's better the ile ingine ni mbaya. Ikiwa kuta inaandika hivyo, I think it's better. So glycoproteins, your spelling, and secretions. Role of mitochondria in a function for respiration. So what is the adaptation of mitochondria to its function? So your assignment. Adaptation and assignment. Adaptation of mitochondria to its function. Adaptations of mitochondria to its function. Now another assignment, and make sure you remind me of this assignment. So in tomorrow's maths class, you can also bring unikumbushe your assignment. Haya. Ama ulize swali kama utakwenda kushinda. On which tissue, on which animal tissue, ama tissue of a mammal are there high concentration of mitochondria? Can you just assess where are mitochondria highly concentrated on which tissue? Then assignment. Alafu ya mwisho, how does surface area to volume, how does surface area ama how do the following factors affect the rate of diffusion? How do the following factors affect the rate of diffusion? Ya kwanza, temperature. Ya pili, surface area to volume ratio. Yeah, so that is important. I think you can also write this question. Where in animals and plants, the following physiological processes, the following physiological processes, where in animals and plants, the following osmosis. So the following physiological processes occur. The first one is osmosis. The second is diffusion. Na ya tatu is active transport. Diffusion, then active transport. Diffusion, then active transport. So we have come to the end of our class today. I want you to do that assignment. You might be surprised to see it in the kata. I want you to do that assignment. So we have come to the end of our class today. I hope you have noted the assignment. Do the assignment. If you have a question, ask me. You can also submit to me formatting. Thank you. See you in the next video. And class tomorrow, we have a mathematics class in the morning for Form 1. Yes.